Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm very excited to be here today with Dr. Vincent Dion, who's group leader at the UK Dementia Research Institute at Cardiff University. His work is at the intersection of DNA repair and neurodegeneration, with a particular focus on repeat expansion disorders. We're going to talk about Huntington's disease. We're going to talk about a number of other important repeat expansion disorders. And what I think is interesting about how the field is evolving right now is within the last couple of years, I would say we've learned some pretty fundamental things about the nature of the underlying biology of these diseases in terms of somatic evolution of repeat expansions. And we're going to talk about this. We're going to talk about therapeutic implications and a lot more. So Vincent, thank you so much for taking the time to join me.
Yeah, thank you for having me. It's quite exciting to be here.
I want to start actually with where you started your career. You were in the lab, I think, of Stanley Miller. So you worked on origins of life. I'd love to actually just learn a little bit about what that was like, what you were working on, because it's for anybody in biology, it's just one of those fascinating areas to think about.
Yeah, that's really sort of how I got into science in the first place. Sort of thinking about, what this is all about, where we come from. And so this is back in the late 90s, right? So so I guess it was 1998 where the internet was relatively new. And I saw a program, like a summer internship, basically advertised out of UCSD. And I said, I'm just going to apply for this. And somehow I got it and got in the lab of Stanley Miller, who, so for those of you who don't know, is the guy in the 50s who put in a sealed glass container, he put some water, boiled it, and he also had some ammonia, I think, it was ammonia, it was hydrogen, it was something else, I can't remember now. And then you would zap it with electricity and boil the water, and at the end of the week, you end up with amino acids, right? So the idea was to simulate sort of a prebiotic, chemistry. And so what I did that for 2 summers in 99 and in 2000, I think. And it was about basically the question of why is it that DNA has picked cytosine and thymine as the pyrimidines and not, uracil and cytosine instead, or 5-methylcytosine, and the idea was that it had to do with the stability of the 5 substituted pyrimidines. And so I ended up, sealing solutions of 5 substituted pyrimidines into glass jars and then doing HPLC for 2 summers. And it was a ton of fun. It was my first lab experience. And I was just really lucky to be there.
Yeah, that is very cool. How did you then get into genetics? Where, you sounds like you got into genetics in a very foundational sense there, but medical genetics, Where was your first introduction?
Well, I sort of never really planned to do any medical genetics to speak of. I think that's a theme really. So what I ended up doing was I had convinced myself that I wanted to do molecular biology. Did I really know what it was? Not really. But I just sort of didn't ask myself too many questions and went for it. And, with the 5 substituted pyrimidines, I had a rough idea of what DNA was about. And, that led me to mutations. And that's sort of like, if I look back, you can see the I can see the path was sort of not completely obscure, but so from there, so I did an undergrad in molecular biology at the University of Guelph. And from there, I wanted to do mutagenesis in bacteria. And because I just specifically stress-induced mutagenesis in bacteria, which I thought was really interesting because obviously, usually with evolution, you get, a variation in the population and that gets selected, right? Whereas the stress-induced mutations, it was mutations occurring in response to the change in environment. So I thought conceptually that was really interesting. And so I went to Houston to Baylor College of Medicine to do this. And with the rotations that they have in the US, then I ended up in a lab that was working on repeats in mammalian systems. And it was just a really interesting DNA repair problem. And so I stuck to that, changed my mind, and continued. And I haven't really left the field since. So that's been sort of the path to, and the repeats, and then led me to more disease relevant and translational research, but that's much more recently.
Yeah, can you, I very briefly mentioned some of the recent developments in our understanding of Huntington's disease and more broadly, so you also, I think, work on myotonic dystrophy, there's some spinocerebellar ataxias that have important repeat expansions. Let's start with Huntington's maybe. For people who aren't super familiar with it, could you talk a little bit about the repeat expansion in general and also some of the recent work on how the somatic expansion drives the phenotype in the brain?
Yeah, so basically so Huntington's research as we know it today started by the cloning of the gene in 93, finding an unusual mutation, which is a repetitive sequence. It's three bases, so C, A, and G, at the same locus that expands in the patients beyond about 36 repeats in that case. And there was this observation from the very beginning that the longer repeat tracks were leading to an earlier age of disease onset, but the numbers were quite low and people didn't pay so much attention to this. In the meantime, there was the observation that these repeats were unstable. So if you did an old-fashioned Southern blot, what you see is a smear, so suggesting somatic mosaicism. And that turns out that's because of the repeat size changes over time and bias towards expansions. And so there's a bunch of people, including myself, that started working on sort of the DNA repair aspect of this, which is basically the really interesting part, I think, is that DNA repair, which we tend to think of as protecting the genomes against mutations, is actually actively making mistakes on this aberrant substrate. But again, people working on the sort of the pathology of Huntington's sort of didn't view this as an obscure branch of DNA repair that was not really relevant to anything.
Right.
I remember in 2006, I was a PhD student at the time, and Christopher Pearson sort of polled an audience at a conference to see whether they thought somatic instability had anything to do with the pathology or not. And, you have like a handful of hands going up. And then fast forward to 2015, and it's the first GWAS that comes out. And then it was the more detailed one with more numbers came out in 2019. And this is really where the idea that DNA repair, specifically mismatch repair, was a modifier of the age of disease onset beyond just the size of the inherited repeat. And that really sets the stage to what we're looking at now, which is, people trying to target the DNA repair pathways that drive somatic expansion to try to prevent it. And so that's really where the field is at the moment. We're really in an exciting time where there's a lot of ways of trying to, there's a lot of opportunities basically right now, like new inhibitors of DNA repair, but also repurposing things that are typically used in the context of cancer that we could potentially used in the context of expanded repeat disorders. We've been doing gene editing as well. I'm guessing we're going to talk about this next. And so there's a lot of different approaches at the minute that really aim at the DNA, really, the mutation itself, that's dynamic. And I think that's really interesting. It's really unique.
Let's talk about gene editing, because based on your description there, it sounds like you can obviously go directly at the repeat expanded molecule and try to reduce it in size or take it away with RNA silencing or something like that. But you could also go at one of these DNA mismatch repair genes as well. Can you talk a little bit about some of the therapeutic strategies that are being tested?
Yeah, so the one that's probably furthest along is silencing of the mutated Huntington copy. And so the idea is you knock it down, you have less of the toxic species, you have less neurodegeneration. So that conceptually is fairly straightforward. What's happened is that there was a trial by Roche a couple of years ago that was discontinued because, well, people that were receiving the ASO against Huntington were doing worse than the placebo. And so that was terminated early. And that was the first problem. So, and then from that came out the idea. And of course, I should say it was terminated early. It was technically a failure, but there's a giant amount of data that came out of it and expertise, and it facilitates what comes next after that, right? Yeah. But one of the things that came out was this idea that you needed to have an allele-specific treatment. So Huntington's is caused by the expansion of a CAG repeat, but you only need one of the two alleles to be mutated to show the symptoms, right? And so what that means is if you have an allele-selective drug, it will only decrease the amount of the mutated Huntington and not of the non-expanded allele. And that's an advantage, the hypothesis goes, because you're not lowering all of the Huntington protein, you're only lowering the one that is mutated, leaving the one that's does the normal function to do its job and to prevent any disease state. And so that's been sort of what came out of this. But it takes there's a big lag between the development of these allele-specific approaches and the clinic. So we're still so uniQure recently has had some success as well. They're also silencing, but using a very different approach. They're using AAVs injected directly in the brain that had seemed to be working better than the ASOs, but it has the drawback that in terms of scalability, and there's potential problems with knocking down both copies of Huntington.
And that one, just to interrupt, am I right? The way that one works is so it's delivering, like you said, it's delivering an AAV-based gene therapy, and I think it's producing a microRNA, right? And so that microRNA is then going in and knocking down just the mutant allele of HTT.
It's both actually in that case. Still, there's some more in preclinical development that's allele specific. Some of them have been for myotonic dystrophy, for instance, have been starting to be tested in the clinic as well.
Really interesting.
But yeah, so this sort of one general approach, so silencing Huntington. There's a second approach that's the splicing modulators, and those are small molecules that would basically mess with the splicing of the mutated allele. And so those are also in development. There's a few different ones around. Again, the idea being you reduce the amount of, and those would be allele selective, ideally. And so this would be another way of reducing the mutant Huntington. Then there's the whole somatic expansion that we were talking about. So the idea there is to have a small molecule inhibitor. So that would be very convenient, taking orally, that would distribute throughout the body and prevent somatic expansions from happening. And this, in theory, would delay perhaps to the point of preventing altogether the appearance of the symptoms. The drawback in that case is that we're looking at a very long-term treatment. So the uniQure, for example, is a one-off surgery. Yeah. It's invasive, but it's going to be done once. The small molecules is forever. And with the potential of targets that, but even just the potential on targets, right? We're targeting the DNA repair pathway that, and so in the long term, is that going to have a negative effect? We don't know yet, and we will only find out after trying it out.
Yeah.
And so I think there's a lot of optimism around that, in that, and people are really hoping that there's not going to be any side effects. There's some data suggesting that there won't be, but I think the jury's still out. And of course, we all have our own biases. I'm very much on the gene therapy side of things. And what I'm trying to do in my lab is to really correct the mutation that causes the disease. So we're not knocking down HTT. We have an allele selective approach that was not designed. It just happened to be that way by chance. And so that would not decrease the Huntington locus. It would be a little selective. It would be a one-off permanent correction of the mutation. And so this is sort of where we're trying to go towards. Obviously, there's going to be some drawbacks for this as well. The same ones because it's a neurosurgery, but I think it might be better to correct the mutation than to simply silence it.
And so what is the healthy range? Are you just going in, and maybe I'm oversimplifying this, but just removing a big chunk of repeats to get it to a healthy range? Is that the right way to think about it?
Yeah. So, I'm going to make it more complicated.
That is fine. That's what I'm here for.
So the early approaches that people had for gene editing of repeats is exactly that. You cut it out and you're done, right? So the problem with those is that they're not allele selective, because your Cas9 that's cutting on the outside of the repeat will be cutting both alleles, unless you have a SNP, in which case you're looking at a subset of patients from a rare disease, and that starts to be very incredibly personalized and expensive. So what we ran into doing completely other things, not designing a gene therapy approach, was to ask a very basic question, which is, what happens to repeat instability if you deliver different types of damage within the repeat track? And so at the time where we started, Cas9 had just gotten online, so CRISPR, and we could cut the repeats, induce a double-strand break. Or what we could do is point a point mutation in the Cas9 enzyme, turn it to a nickase, which nicks only one of the two strands of the DNA, and then see what the outcome was on the instability. And what we found there really would set us up towards the translation is that a double-strand break, you'll end up with expansions and contractions, which in terms of gene editing is not fantastic. Plus, there's a lot of repeats in the genome that would be targets, not a good idea.
Right.
But with a nickase, we ended up with mostly, if not entirely, contractions. And so obviously when I saw this, we were like, we're working on something different here all of a sudden. And it's taking us a while to sort of go from cell lines for basic mechanism to a mouse model and making AAVs to work. And like I had never done this before. It's but we followed the results again, and this is where it's leading us. So by now we have a preprint right this moment online where we can show that we can correct the mutations in stem cell derived neurons and astrocytes from Huntington's. We can do this in myotonic dystrophy neurons. It works in the mouse brain, and that's accompanied by improvements at the molecular and behavioral levels in one mouse model. So I mean, I say this, and if you would have told me this 10 years ago, and I would have been like, that's nuts. It's taking you so long. What happened? But it's never the things that you think won't, like the things that haven't worked have not been the things I would have predicted wouldn't work. Like it seemed to be the obvious, but you know.
Oh, interesting. What were the things for those that are maybe early on in that journey right now? What are the unexpected things that slowed you down?
Yeah, so I think the first mice that we injected with AAVs was in I want to say 2016, maybe 2017. And we were getting no Cas9 expression at all in vivo. Also, no contractions, but we couldn't tell whether it was because of the lack of expression or because of the fact that it didn't work in neurons or something. And it took us a long time to realize that Cas9 in the brain ends up being silenced fairly quickly. And so if you're doing regular gene editing, then people would just look at the DNA and see the edits and go like, well, it's been there, we're done. But nobody had really done any staining of Cas9 in the brain. And the reason for that is that it goes away after about 2 weeks. And so, and with the nickase, the disadvantage is that we need a long-term expression. These contractions are relatively rare events, and we need Cas9 to be present for weeks and weeks. And so it turns out that as we tried more and more constructs, the key has been to do not a species-specific codon optimization, but a cell-type-specific human codon optimization. Again, like this is not something that I would have expected that we would have needed. Why? But this was the main obstacle to us being able to show that we can actually get contractions in vivo. And that will be, I suspect that will be the limiting factor to applying it to multiple different diseases as well, because we'll need to adjust the constructs for every relevant tissue, depending on the disease. And so this is going to be rate limiting for sure.
And when you say to the tissue, is that down to to the striatal neurons versus some other brain cell type, or is it more like if you wanted to go after myotonic dystrophy, then you need to adjust it to get into muscle cells?
Yeah, so we're not completely certain yet. We stumble on this by chance where, and it's a little bit of chance, it's a little bit of we have a construct that's expressing, let's just go for it. And I think like for what we're using in the mice at the moment is a muscle codon optimized. And the way it was designed is to take the top 10 most expressed genes in the muscles, look at, codon frequencies and use that in a Cas9 construct. And that's really helped boosting the expression. Why is it that the muscle codon optimized works better in the neurons? I don't know. Does it bother me a little bit? Yes. I would like to know, but it's not a lot of difference. Like it's 16% of the codons that were changed, and we're not changing the amino acids. We're just changing the codon. There's no splicing in there, so it seems to be a translational effect, co-translational effect. It's really quite interesting, but
yeah. And just to make sure I'm getting it from a mechanistic perspective, what you think is happening with the Cas9 nickase is, so normally in the brain, these CAG repeats, they form very unstable loops, right? And instead of you mentioned earlier how there's a mismatch repair issue. Basically, instead of fixing it, additional repeats are created and you have this expansion cascade. But is what's happening with the single strand break is it almost reverses that issue, where then it does see there's an issue here and it starts removing repeats instead of adding them. Is that about right?
Yeah, so what we think is going on is that the nickase, because the guide RNA that we're using targets the repeat track itself, will end up with, and this is me speculating, by the way, like this is sort of the working model.
Yeah.
But so what we think is happening is that we have multiple nicks on the same strand of the DNA, and that will create, that's processed to create a single-stranded DNA gap. And that allows, anytime you have these repeats that are exposed to single-stranded DNA, they form into these non-BDNA structures, hairpins, loops, whatnot. And that is why they're unstable, right? So these are the structures that are really recognized by the repair machinery. So the idea is that we would end up with one strand that has, that's been religated, and one strand that still has the loop. And then that can be processed further by endonucleases, or in the context of replicating tissue, it could just be replicated. And then you would end up with a contraction. And so this is sort of how we're thinking about it. And that explains the allele selectivity, because if the repeat track is too short, then you don't get the single-stranded DNA gap. Yep. You don't get a stable structure. And so it's not unstable. And in unpublished work, what we're finding is that the mechanism, so the genetic requirements for the contractions by the nickase are very different than what you expect to see by with somatic expansions. And so basically we're going to have a lot of fun trying to figure out what the mechanism of contraction is. And yeah, like my basic DNA repair brain is going to enjoy itself with that.
Yeah. Sure. Yeah, it's challenging, but it is kind of fun when you get these very unexpected results, right? Because it gives you something to really really chew on for a long time, right? And it will change the way that you think.
Oh yeah, for sure. Yeah. I mean, it's, I think a really fun part of science is that it brings you places you didn't expect to go. And if you dare to go, then you find yourself in more places you never expected to be. We're talking about how to design, a first in human trial. And it's not like I thought I have any specific knowledge. Like I need the expertise of people around to get this done.
But I was actually just literally going to ask you about that, because you can keep following this down the path to understand the mechanism in a more fundamental way, but then you can also take this and take it from the mouse and stem cell models into human. How are you thinking about that big leap?
So I'm thinking about doing both at the same time.
Yeah.
So I'm very lucky to be supported right now by the UK Dementia Research Institute, right? And so that has allowed us to do some long-term planning to do some of those troubleshooting that's taken for a long time. And so that's been a real advantage. I don't want to, minimize that. And the other aspect of this is I feel like if I'm not pushing this towards translation, it's not that other people will suddenly jump on this, right? And so I feel like I need to give it a chance to succeed, and for that I need to find people who I can convince that this is a good idea, then we should try and move forward. Yes. And luckily, Cardiff in particular has been really great for this. We were one of the sites for uniQure trials. So there's a lot of expertise in the neurosurgery, in the neurology. So the HD clinic in South Wales is here in the building. And so there's a lot of interaction with the clinicians that are here. And so that really is something that I couldn't have done in most other places in the world. And so I'm finding myself in a position where we can, and that I feel like I should. And so why not, the decision then becomes fairly easy to just try to get it done. Yeah. And on the mechanistic side, it's a much more fundamental question. And I think it's a very good academic question that can be followed with, grant applications and that kind of stuff, right? And so this is sort of we're trying to see if we can go the spin out direction for the translation and remain in academia for the mechanism of action.
We've talked about uniQure, and for those who aren't familiar, they have been in the news recently because they had what I thought was really very strong data in their Huntington's trial. They had an external control arm, and they also had a randomized control trial, but the FDA has publicly been asking for more and additional data, particularly a placebo-controlled trial, I think, which is incredibly difficult to do. How do you think about that in relation? It's definitely makes something like this feel like an uphill battle.
Yeah, I mean, so I think there's a couple of different avenues to look at this. Like, one is just ethically speaking, in Europe, you won't have a sham control for a neurosurgery. Like, that's just not going to happen.
Yeah.
In the US, it is possible, and there were a few patients that were that had that, where basically they had the anesthetic, a hole drilled in their skull, and then it was patched back up, and that's what they had. So one of the real issues is how you select your patient populations and your historical controls as well at the same time. And I think this is maybe where more could be done here to try to see whether is it that the patient population in one way or another was that received the treatment were different in any way from the historical controls. The other thing to keep in mind is that because the historical controls, you have a lot more of them, so the variability between them is much lower as a result. Whereas here you're subsampling 29 people for injections, and so there's a lot of these sort of study design. I don't necessarily want to say flaws, but the imperfections that I think has led to the FDA reversal of opinion on this. It's not. I don't think it's unsurmountable. I think it just needs to. You have to keep in mind as well that there's relatively few people that have received the treatment right now, and that one solution might just be that we need to have a few more numbers and see whether that continues to go the way it goes. I think there's also the sense that it's a slowdown, which is great because there's nothing else right now. But given that it's a neurosurgery, a slowdown is that going to be worth it for hundreds and hundreds of people, right? If not thousands?
Yeah.
So I think that there's all of this together. And the one thing I should say, though, is that we don't know what's going to work and what's not going to work, right? And I think from a patient's point of view, like from a single company's point of view, right, you want your own treatment to work for obvious reasons. But from a patient's point of view, it doesn't matter that much what works so long as there's something that does work, right? And I think the best thing we can do is provide options and see which one makes it. And the first one won't be perfect. It's going to be a few treatments down the road where we're going to get the hang of it.
Yeah,
that's something to keep in mind.
Yeah, that's right. We've been talking a lot about DNA editing, but I think you are also doing work on epigenetic editing, epigenome editing. Can you talk a little bit about that concept and what you're working on?
Yeah, so we've moved away a bit from epigenetics and DNA methylation, simply because I had to pick a lane at some point. I couldn't do everything, and then the nickase work was working so well that we decided to go down that route. Historically, I've been very much interested in sort of the interaction between the local chromatin structure and the instability of those repeats. There's quite a bit of, and the real challenge there is the fact that with this, it tends to be a correlation. You tend to correlate sort of the epigenetic state and the chromatin structure to the instability of the repeats. And getting the sort of the cause-effect relationship is not trivial. And I think that is probably why, we had the really successful sort of gene editing approach, and we went down that road instead. It's not that I'm not interested anymore. It's just that there's just so many hours in a day.
Yes.
And you have to make decisions. But yeah.
Do you ever stop and just marvel at how the explosion of Cas enzymes and CRISPR in general, and how it came from blue skies research that had nothing to do with therapeutics, right? And it's blossomed into something that none of us could have predicted 15 years ago.
I mean, yeah, and there's a lot of examples of that, right? So a lot of the times the basic research, I mean, it's just like venture capitalist, really, where, you're going to have to invest in a lot of different things before one of them sort of takes off and really is the next Google. And so it's the same thing with biological tools and approaches. You need to, invest in a lot of stuff that goes nowhere. That's just interesting for the sake of the biology, for it to suddenly start becoming really revolutionary. And I think Cas9 is one of those, right? And I think nobody really wakes up in one morning and says, well, I'm going to now invent a completely new way of editing DNA, right? That's not really how things work, right? And I think is the biology will dictate what the applications might be down the line.
Yeah.
I think it
is humbling in a way to know that in some ways it's so hard to predict the future, because whatever we're going to be doing in 20 years, there's probably some kind of basic science breakthrough right now that by definition none of us are thinking about, that's going to bubble to the surface, and it's going to change a lot very quickly.
Yeah. No, I mean, for sure. I think that one of the first examples was restriction enzymes, right? And in some ways, now they're obsolete. You don't need them so much anymore.
Yeah.
And it's been replaced by Cas9. It's been replaced by, direct sequence synthesis of DNA. And it's like the progress has been like, I'm in touch with my PhD advisor still. He's been retired for a bit. And, when he started he didn't have a computer for the longest time. Actually, when I did my first internship, well, both my summer internships, my Stanley Miller had no computer in his office, right? So no typewriter. His secretary would print out his emails. He would write down by hand the reply, and she would type them up and send them out. Right? So it's not, I mean, it's a long time ago, but it's one generation ago, basically.
Yeah.
What it's changed like next.
Right? Very quickly. We'll probably all have AI assistants that do some version of all of that stuff that isn't thinking about the deep scientific problems, and they'll probably help you with that too.
I hope so. I definitely hope so.
One of the things I often like to do towards the end of the episode, especially once we've covered a lot of the sciences, is something outside of the science. And something that you'd mentioned before is you have something that is pretty common with with couples these days, which is what's called a two-body problem. So you have a partner that is, I think, also an academic, right? And so we're probably many people listening to this podcast have a similar challenge, which is you got two people who love each other and are trying to figure out how they make their careers work, especially in depending on what field you're in. It can be geographic constraints where there's only particular places people want to be. What have you learned about making that part of your life successful?
I've learned that the university systems are not meant for two-body problems. I think it also creates opportunities, right? So the reason why we ended up, my wife and I, going to Switzerland for postdocs is because she's Turkish. We met in Houston, and she said, we're going to Europe. And I said, okay. And so, it's, but what's interesting, I think, as well, from because when we were looking for jobs, they would talk to her and they would think I'm a trailing spouse, and then they would talk to me and they would assume that she's a trailing spouse. And in our particular situation, we were about the same level, very close to each other anyway. And that created a problem, because people were starting to sort of be cued in the idea that maybe a trailing spouse is easy to accommodate or easier. But someone who needs a lab, that's a lot more challenging. And I think that's one of the reasons why we ended up in Cardiff, because we were at the right place at the right time. But I think there needs to be people in power needs to be thinking about this a lot more. It's very good for diversity because once, if you have a couple, it also, by definition, it's going to be 50% women, 50% men, or on average. And then what will happen is that they're not going to be leaving or going anywhere.
Yeah, you get a lot more, probably loyalty as well, just if an institution can figure out how to make it work for you, right?
Yeah, exactly. So, and I think funding bodies have not clued in on that at all either. I mean, it might just be that it's a niche problem, but yeah, and everybody's got their own constraints on finding an academic position. It's one person is always difficult. And then if you add regional constraint or geographical constraints or two-body problems, then it completely changes the, on the plus side, we interviewed everywhere in Europe, everywhere in the US and Canada. And yeah, it takes a lot of time, but you end up making it work.
Yeah, probably at minimum, you just have to do way more applications, right? You just have to put a lot more opportunities in the top of the funnel versus if you're doing it by yourself.
Yeah, and I think often I would reach out and say, "Hey, there are two of us. Is it worth my time, really?" Yeah. And people are reluctant to say, "Oh, no, forget about this. We're not going to be able to accommodate this." Or, And maybe one way is just saying look, I can't tell you whether you should apply or not, but there's no real mechanism to be doing this, right? And you can be lucky, but maybe not.
Yeah.
And I think, we've run into a few stories that where they were my wife already had one example. My wife already had a lab. We were looking for tenured positions, and they wanted to interview me, and then we had said, there are two of us, and the reply we got is we're offering her a postdoc position in someone else's lab, right? And that's incredibly insulting. And so somehow we didn't end up doing that.
Is your wife also in biology or a different field?
Yeah, so she's doing cell biology, developmental cell biology, basically, is what she's interested in, organ size in particular. So it's very fundamental. So we've been on the same department before. Right now, we're not. So, whether that's easier or not.
Yeah.
It's not clear.
Probably depends. Very cool. As we wrap up here, we talked about really the two big directions that I think you're taking: first in human, then also really understanding this mechanism. Is there anything else that that's plenty, by the way, but is there anything else that you're excited about right now that you want to leave people with as we close out?
I think maybe more of a concept is, and that is, there's a lot of things that could be done around monitoring efficacy of the treatments that target DNA. And so biomarkers, but around, how do you measure target engagement, which would be sort of the technical in the brain for these, and I don't know what the answer is, right? But I think that's somewhere that I'm planning on investing some thought into, because that's going to be really important for clinical trials to be successful, regardless of what the approach is. We'll need to be able to do that.
Yeah, that is really interesting. How would you do it today? Where are we starting from for something like this?
Well, so if you take the uniQure trial, for example, or some of the ASO trials, what typically for Huntington, what people will look at is blood biomarkers and CSF, so cerebrospinal fluid. So proteins, for example, so mutant Huntington in those in the CSF, or neurofilament NFL, neurofilament light chain in the blood, but they're not super specific, and they're not very widely applicable. Like you won't take the NFL in the blood for myotonic dystrophy, right?
Yeah.
And so this is sort of how it's done right now. But I think there, like people have done, functional assays on extracts, for example, that might also help. There's a lot of different ways of thinking about this, but it's challenging to go from the lab and cells into patient tissues. And I think that leap is going to be also important to be making. But yeah, biomarkers, but from a sort of a, how do you, so it's not biomarkers of disease progression per se, it's biomarkers of whether a treatment targeting DNA works is basically where my brain wants to go.
Yeah, that's interesting, and maybe thought-provoking for somebody who's thinking about the same thing.
Yeah, well, I hope so. I mean, yeah, I could talk about it some more, but I probably shouldn't. I should be writing grants instead.
Well, Vincent, this was really great. Thank you. I think we covered a ton of ground. You're doing really cool and impactful work for patients. So thanks for taking the time to share it with us.
Yeah, thanks. It's been a pleasure. Very fun conversation.
Yeah, the pleasure is mine. And everybody, thank you as always for listening to this episode, and we'll see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.